I welcome all the registered students in the SWAM course of Genetic Engineering and Recent Advancement. In previous lecture, you have been gone through the topic steps involved in genetic engineering. The topic that we will cover in today's lecture is principle of cloning. I am Dr. Kush Kumar Nayak, Assistant Professor, Amity Institute of Biotechnology, Amity University, Chhattisgarh, is going to cover the following content in today's lecture. We will cover introduction, definition of cloning, historical background, types of cloning like reproductive, therapeutic and molecular cloning. Then we will see cloning techniques like nuclear transfer cloning, embryo splitting cloning. Then we also see advantage and disadvantage of cloning, its applications in the field of agriculture, medical and research areas. Then we will discuss the concern in human cloning, then legal restriction involved with human cloning. Then we will also see cloning and genetic engineering, the relations in between two, and then discuss challenges and limitation. We will also see certain important cloning breakthroughs, cloning myth and misconceptions we will discuss in this lecture, and then we also discuss the future of cloning. So, let us start with the introduction of cloning. Here cloning is a scientific process that involves creating identical or genetically similar copies of an organism. It is technique that have been extensively studied and applied in a various fields including agriculture, medicine and research field. The concept of cloning dates back ancient time, which early example of cloning observed in plants through asexual reproduction. However, it was not until the 20th century that significant advancement in cloning technology were made. As we know, cloning refers to the process of producing genetically identical copy of an organism, cell or a DNA fragment. This can be achieved through various techniques like reproductive cloning, therapeutic cloning and molecular cloning types. As we know, reproductive cloning involves creation of genetically identical organism by transferring the nucleus of an adult cell into enucleated egg, while therapeutic cloning on the other hand aims to generate embryonic stem cells for medical research and potential treatment. And molecular cloning involves the replication of a DNA fragment and their insertion into the host organism, often used in genetic engineering and biotechnology areas. So, cloning has been a topic of debate and ethical concern due to its implication and potential risk. However, it has also offered several advantages such as the ability of reproduce endangered species, develop genetically superior livestock and crops and potential advanced medical treatments. As the field of cloning continues to progress, further research and ethical consideration are essential for responsible and beneficial application of cloning technology. So, before moving forward, let us understand the historical background. The concept of cloning has rich history that dates back in ancient time. Although the term cloning was coined much later, early example of cloning can be observed in nature through asexual reproduction in plant, such as budding and vegetable propagation. However, it was not until 20th century that significant advancement in cloning technology were made, leading to the birth of modern cloning technology. One major milestone in cloning history is the development of nuclear transfer in the 1950s. British biologist John Gordon conducted an experiment where he successfully transferred the nucleus of a frog intestinal cell into enucleated frog egg, resulting in the development of a cloned tadpole. This experiment provided significant insights into the potential for genetic reprogramming and possible of cloning animals. Another critical breakthrough in cloning history was successfully done in mammal Dolly the sheep in 1996. Scientists the Roslin Institute in Scotland, led by Dr. Ian Wilmot, used nuclear transfer to create an exact genetic replica of a sheep. Dolly birth marks first successful mammal cloning, capturing the attention of the scientific community and general public. Since Dolly's creation, cloning technology has continued to progress rapidly. Researchers have successfully cloned various animals including cat, dog, horses, mice and pigs. Each cloning achievement has expanded our understanding of the principle and techniques involved in cloning, pushing the boundaries of what is considered possible. The historical background of cloning is not limited to animal cloning. In the scope of plant cloning, techniques such as tissue culture, plant propagation have been utilized for centuries to produce identical copy of plants. These methods paved the way for modern plant cloning techniques, which involves manipulation of plant cells, tissues, hormones to generate clone plant with desired traits. The historical development of cloning have been marked by both significant advancement and ethical dilemmas. The cloning of animal and potential application in human cloning continue to raise complex questions regarding ethics, morality and implication of manipulating life at such a fundamental level. As the field progresses, further research, exploration and ethical consideration are essential to ensure responsible and beneficial application of cloning technology. So, let us try to understand various types of cloning. The differences between three types of cloning are we can consider it in three category. One, reproductive cloning, second, therapeutic cloning and third one is a molecular cloning. Reproductive cloning refers to the creation of identical copy of our organism. Therapeutic cloning, it also known as a embryo cloning or a research cloning. Therapeutic cloning aims to generate embryonic stem cell for medical research and potential treatment. On the other hand, molecular cloning, also known as a DNA cloning or gene cloning, involves the replication and insertion of a DNA fragment into the host organism. Each type of cloning offers unique application and raises different ethical consideration. Reproductive cloning has sparked controversy due to concern about ethical application of creating genetically identical organism. While therapeutic cloning has potential benefit in medical research, but raise concern about the destruction of embryos. And molecular cloning on the other hand is widely accepted as a valuable tool in biotechnology and genetic engineering. The ethical consideration and regulations surrounding each type of cloning vary across the countries and are subject on ongoing debate and discussion. So, let us understand each cloning method in details. First, start with the reproductive cloning. As we know, the reproductive cloning in this process involves transferring the nucleus of a somatic cell from the donor organism into egg that has had its nucleus removed. The egg, now containing the genetic material of a donor organism, is then stimulated to develop into an embryo and implanted into surrogate mother for gestation purpose. The resulting offspring will be genetically identical to donor organism, and cloning of Dolly Ship in 1996 is an classical example of reproductive cloning. The second cloning method, that is therapeutic cloning. In this process, nucleus of a somatic cell transferred into enucleated egg, similar to the reproductive cloning way. However, instead of allowing the embryo to develop into the fetus itself, it is maintained in the laboratory for few days and embryonic stem cells are extracted and cultured. These stem cells have potential to differentiate into various cell types, providing researchers with a valuable tool for studying diseases, study new drugs and potentially treating degenerative conditions. So, the third type, which is known as a molecular cloning, in this technique, which is widely used in genetic engineering and biotechnology, it enables the production of multiple identical copy of a specific DNA fragment, such as gene of interest. The cloned DNA can be introduced into bacteria, yeast or other host cells, where it replicate and produce potential protein encoded by the DNA fragment. While molecular cloning has numerous applications, including the production of a recombinant protein, the study of gene function and the creation of genetically modified organisms. Molecular cloning is a set of methods which are used to insert recombinant DNA into vector, a carrier of a DNA molecule that replicate recombinant DNA fragment into the host organism. The DNA fragment, which may be a gene, can be isolated from a prokaryotic or eukaryotic system. Further, let us try to understand the different type of techniques that have been used to perform the cloning things. So, there overall two main cloning techniques are available here. First one known as a nuclear transfer and second one is known as a embryo splitting cloning technique. In nuclear transfer cloning techniques, it is sometimes referred as a somatic cell nuclear transfer technique, is a technique or is a method used in both reproductive and a therapeutic cloning. It involves transfer of a nucleus from a donor somatic cells into enucleated egg. While in embryo splitting, which also known as a blastomeres separation or embryo twinning, is a technique preliminarily used in reproductive cloning. It typically involves the splitting of a pre-implantation embryo into multiple embryo, each of which has the potential to develop into genetically identical organism. Both nuclear transfer cloning and embryo splitting cloning have contributed significantly to our understanding of cloning techniques and their potential applications. These techniques enables the creation of genetically identical organism, providing valuable tool for research, preservation of endangered species and potential advancement in medical treatment. However, they also raise ethical concern, particularly in regards to human cloning and manipulation of life at a fundamental level. Moving towards to understand the steps that involved in different cloning techniques. The steps involved in nuclear transfer cloning are considered in several categories, which start with the donor cell collection. Somatic cells collected from the animal or organism to be cloned. These cells can be obtained from various tissues such as skin or muscles. The egg typically obtained from the female animal. Next step, where it carefully surgically enucleated to remove its nucleus. This create an empty egg with no genetic material. In the third step, we transfer nuclear things inside the enucleated egg. The nucleus from somatic cell is extracted and transferred into the enucleated egg. This can be done through a variety of method, including microinjection or cell fusions method. The next step, stimulating development will done. This is about reconstructed egg now containing the nucleus from the donor somatic cell is stimulated to begin development. This can be achieved through chemical or electrical stimulation. In the fifth step, embryo then implantation done, where developing embryo is then transferred into a surrogate mother uterus for the gestation and normal development. If it is successful, a genetically identical offspring will be born. Nuclear transfer cloning has been used to clone various animals, including sheep, cows and mice. It has also been attempted in non-human primates and holds potential for a human reproductive cloning, although this remains highly controversial and ethically debated. On the other hand, in embryo splitting cloning, the steps involved here are actually started with the embryo collections, where multiple embryos are collected through the in vitro fertilization or other assisted reproductive techniques, which will then corrected embryo are carefully separated into 2 or more parts, typically at the 4 to 8 cell stages for development. This can be done through mechanical or chemical means, after which each separated embryo is placed in a suitable culture medium and nurtured to develop independently. Each embryo has the potential to develop into a complete organism. The embryo splitting cloning has been successfully done in various mammals, including cattle, pigs and rabbits. However, it is less commonly used compared to the nuclear transfer cloning due to certain limitations, such as potential for epigenetic abnormalities and lower success rates. In the continuation, let us try to understand the advancement and disadvantagements of the cloning aspects. Advancements of cloning we can classify in several categories like reproductive of endangered species. Cloning offers the potential to reproduce endangered species, helping to preserve biodiversity and prevent species extinction. Another advancement is improved agriculture way. Cloning allows for the reproduction of genetically superior livestock and crops with desirable traits such as increased productivity, disease resistance and improved nutritional value. On the other hand, cloning techniques, particularly therapeutic cloning, hold promise of genetic embryonic stem cells for medical research and potential treatment. These stem cells can be used to study disease, develop new drug and potential replace damaged or diseased tissue and organs. Another advantage or application we can say is related to disease research, where cloning animal with a specific genetic disease enable a scientist to study the development and progression of disease, leading to advancement in medical research and the development of treatment. Cloning could potentially provide a source of organ and tissue for transplantation, reducing the damage for donor organ and reducing the demand for donor organ and addressing the shortage of available organ for transplantation. Let us also understand the points related to disadvantage of cloning. Like first disadvantage is a ethical concern. Cloning raises ethical concern particularly in regard or to human cloning, as it involves ethical consideration related to playing with nature, creating identical copy of organism and potential misuse of technology. The success rate of cloning technique, especially in reproductive cloning, can be relatively low. Many cloning attempts do not result in variable offspring, and cloned animal may have health issue or abnormalities, which is another disadvantage of this method. The genetic diversity, where cloning directly replicate the genetic material of an individual, reducing the genetic diversity. Limited genetic diversity can make clone organism more vulnerable to disease and environmental changes. The another disadvantage is epigenetic abnormalities, where cloning can lead to epigenetic abnormalities, which are alteration in a gene expression pattern that can affect the health and development of clone organism. And technological limitations, where current cloning techniques are complex and require specialized equipment and expertise. The cost associated with cloning can be high, limiting its accessibility and widespread use. The field where cloning applications actually happen are categorized in several parts. The first area where it used in widely, that is reproduction of endangered species. Cloning offer the potential to reproduce the endangered species and prevent species extinction. By creating genetically identical copy of endangered animals, cloning can help preserve biodiversity and contribute conservation of efforts. In agriculture, cloning has application by allowing for the reproduction of genetically superior livestock and crops. The ability to clone animal with desirable traits such as increased productivity, disease resistance, improved quality of meat or milk can lead to improved agriculture practices and increased food production. In medical research area, cloning technique, particularly therapeutic cloning, hold promise in medical research. Cloning provide a source of embryonic stem cells, which can be used to study disease, test new drug and potentially develop regenerative therapies for disease and injuries. Cloned animal can also be used to study genetical disease and development treatment. Another area where it can use is animal model for research. Cloning animals with specific genetic characteristics or disease can provide valuable model for studying various disease and medical conditions. These cloned animal can help researchers understand disease mechanism, test potential treatment and accelerate the development of a new therapies. Another area where it can apply is organ transplantation, where cloning offers the potential for generating organs and tissues for transplantation. By cloning an individual's own cells and growing organ from them, there is a reduced risk of organ rejection, as the cloned organ would be genetically identical to individual. This could help address the shortage of a donor organ available for the transplantation purpose. The biotechnology and new drug production is another area where it can use. Cloning play a significant role in biotechnology and drug production. It allows for the production for large quantity of specific protein such as insulin and growth hormones by cloning cells that can produce this protein. This has application in pharmaceutical industry for development of therapeutic protein and vaccines also. Another area where it use is conservation of endangered species, as we discussed in the previous points. It helps to prevent the biodiversity of a particular ecosystem. Cloning provide a way to reintroduce or the preserve species that are on the verge of extinction. So, let us try to understand the concerns that are related with human cloning. Human cloning is a highly controversial issue that evokes ethical, moral and legal concern. Cloning refers to the creation of an organism that is exact genetic replica of another individual. While the idea of human cloning has captivated both scientific and public attention, it has also raised numerous ethical questions that challenge the very essence of what it means to be human. One of the primary ethical concerns surrounding human cloning is the potential for exploitation and commodification. Critics argue that cloning would undermine the intrinsic value of human life by treating human beings as objects to be produced, manipulated and discarded. They fear that it would lead to a designer baby mentality where individuals seeks to create clone with a specific desirable trait, perpetuating social inequalities and discrimination based on a genetic makeup. Another concern is the potential for physical and physiological harm to the cloned individual. Some argue that cloning could lead to increased rate of health issues and abnormalities in the clones, as seen in animal cloning experiments. Additionally, cloned individuals might struggle with identity issues, such they would share the exact genetic makeup of their genetic donor, potentially inhibiting their individuality and sense of self. The ethical concern also extend to the potential violation of a human dignity and autonomy. Cloning involves manipulation of human life and could be seen as an infringement of an individual's right to self-determination and reproduction. It raises questions about the unique bond between parent and their offspring, as clones could be seen as mere replicate rather than individual with their own distinct identity relationship with their progenitors. So try to understand the various legal restrictions that are associated with the human cloning. We will discuss here about the certain legal restriction that we have to keep in our mind when we go through the cloning concepts. Currently, several countries have implemented legal restriction on human cloning. For example, in the United States, federal funding is prohibited human cloning research, and Food and Drug Administration has declared human cloning for reproductive purpose as illegal. However, there is a distinction made between reproductive cloning and therapeutic cloning. While reproductive cloning is widely regarded as unethical and illegal in many countries, the therapeutic cloning is subject to different regulation and may be allowed under strict oversight. Nevertheless, the legal status of human cloning varies globally. Some countries like the United Kingdom allow therapeutic cloning with strict regulation and oversight. Otherwise, like Canada, have banned reproductive cloning explicitly, but do not have universal legislation governing therapeutic cloning. There are also countries such as Germany, Austria that have enacted laws banning all forms of human cloning. So, the ethical concern and legal status of a human cloning continue to be debated and evolved over the time. The scientific and medical community grapples with the challenges of balancing potential benefit with numerous ethical consideration and potential risk associated with human cloning. The public opinion on the matter also varies, also reflecting their personal beliefs, religious views and understanding of science. Conclusion, we can say human cloning is a highly continuous issue that raises significant ethical concern about dignity and autonomy of a human being. The legal status of human cloning varies across different countries, with reproductive cloning genetically regarded as illegal, but distinction made for therapeutic cloning. The ongoing debate surrounding human cloning underscores the need for careful consideration, regulation and ethical reflection to navigate this complex issue responsibly. So, let us try to understand the terms cloning and genetic engineering. These are very connected to each other, but we have to understand both terms with very clarity. Cloning and genetic engineering are the two scientific advancements that have revolutionized the field of biology and sparked numerous debate about their ethical implication. Cloning refers to the process of creating an organism that is genetically identical to another individual. This can be achieved through various techniques such as somatic cell nuclear transfer or advancement in a laboratory procedures like reproductive cloning and therapeutic cloning, as we discussed in previous slides. Reproductive clonings as involve SCNT to create a cloned embryo, which then transferred into the surrogate mother to develop into a full organism. This has been successfully accomplished in various animal species, but remain ethically controversial when it comes to human cloning due to concern about human dignity, individuality and potential for exploration. Therapeutic cloning on the other hand aims to create cloned embryo for research purpose, particularly in the field of regenerative medicine. The stem cell extracted from these embryos can be used to generate tissue or organ for potential transplantation, disease modeling and drug testing. While therapeutic cloning has a promising medical application, it also raises ethical concern about destruction of embryo and the potential slippery slope towards reproductive cloning. On the other hand, genetic engineering involves modifying an organism's genetic material through techniques such as gene editing. This can be done by editing, removing or altering the specific genes to enhance the desired trait or eliminate genetic disorder in particular individuals. The most well known gene editing technique is CRISPR-Cas9 technique, which has made gene editing more accessible and efficient. Genetic engineering has the potential to revolutionize fields like agriculture, medicine, even environmental conservation. It offers possibilities such as disease resistant crops, genetically modified organisms for medical research and potential gene therapies for inherited disease. However, genetic engineering also raises ethical concern, including the potential for unintended consequences on environmental and biodiversity. Disparities in the access to genetic enhancement and the ethical implication of altering the genetic makeup for future generation. Both cloning and genetic engineering have immense potential in various fields, but they also present complex ethical dilemmas that deserve careful consideration and regulation. Balancing scientific advancement with ethical consideration is crucial to ensure responsible and sustainable practices that prioritize the well-being of individuals and society as a whole. So, let us discuss about certain challenges and limitations that are associated with cloning. It is important to note that while cloning offers potential benefit in various areas, there are also ethical consideration and limitation associated with this application. Careful assessment, regulation, ethical practices are necessary to ensure responsible and beneficial use of cloning technology. Cloning, despite its scientific advancement and potential benefit, faces several challenges and limitations that impede its widespread use and acceptance. The first point that is related with its challenge is low success rate. Cloning success rates in mammals, including human, are typically low. It involves a complex process that requires precise manipulation of genetic material and successful development of an embryo. Many attempts result in failed pregnancies, miscarriages and individuals with health issues and abnormalities. Genetic abnormalities is another limitation, where cloned organisms often exhibit genetic abnormalities which can lead to health problems and reduce lifespan. These abnormalities can arise due to error during the cloning process or epigenetic changes that affect gene expression pattern. The risk of abnormalities can higher and utilize of cloning for reproductive purposes. The another concern is related to the ethical way. Cloning relates significant ethical concern, particularly in human cloning, as we discussed in earlier slides. Cloning can challenge and fundamental principle of human dignities and the unique bond between parent and offspring. It also limit the reduced genetic diversity, which means cloning restrict the genetic diversity as it reproduces an exact genetic copy of individual. This lack of diversity can have negative consequences in terms of resilience to disease, adaptability to changing the environment and overall population health. It can increase susceptibility to genetic disorder and reduce the ability to respond the new traits. Another limitation is related to the technological limitation, which means the complex nature of a cloning technique and need for sophisticated laboratory condition limit the scalability and practicability of a cloning procedures. Cloning requires advancement equipment, expertise, resources, making it inaccessible and costly for many institutions and the researchers. The physiological and identity issues is also a part of its limitation, where cloned organisms may face challenges related to their psychological well-being and identity. The knowledge that they are genetical replicate of another individual, potential social stigma and pressure to live up to perceived expectation can create psychological distress and identity confusion. The lack of public acceptance is also a part of challenge related to cloning. Cloning encounters significant public skepticism and apprehension due to the ethical and moral concern associated with it. The fear of playing God and potential misuse of cloning technology often contribute the negative public perception and resistance to its implementation. While cloning has made significant strides in scientific research, this challenge and limitation highlight the need for rigorous ethical consideration, continued scientific exploration and development of alternative technology to address the underlying concern. As the field of progress, it is important to strike a balance between scientific advancement and responsible practice that prioritize the well-being and ethical treatment of an individual and society as a whole. So, let us discuss certain breakthrough that happening in the field of cloning. Over the years, there have been several significant breakthrough and achievement in the field of cloning. These advancements have expanded our knowledge of cloning techniques, contributed medical research and paved the way for potential application in regenerative medicines. The first example is Dolly sheep, the first mammal to be successfully cloned from an adult somatic cells that was born in 1996. This breakthrough, led by researchers at the Roslin Institute of Scotland, demonstrated the feasibility of cloning using somatic cell nuclear transfer. Dolly's birth opened up new possible for cloning in various animal species. Clone a monkey. In 2018, scientists in China successfully cloned two long-tailed macaque monkey using the same SCNT techniques used for Dolly sheep. The cloned monkey named Zhongzhong and Huahua were the first primates to be cloned using this method. This achievement marked a significant milestone in primates cloning and potentially contributed advancement in a biomedical research. Cloned livestock. Cloning techniques have been successfully applied for livestock animals such as cattle, pig, goats for various purposes. In 2009, a cloned bull named Starbuck was born, becoming the first commercial cloned animal in the United States. This breakthrough opened the door to potential improvement in livestock breeding, disease resistance and production efficiency. Therapeutic cloning and stem cell research. Advancement in therapeutic cloning and stem cell area, this technique have allowed for the creation of a cloned embryo for research purpose. Stem cell extracted from this embryo have provided valuable insights into development biology and potential application in regenerative medicines. So, this research could contribute finding treatment for conditions such as spinal cord injury, Parkinson's disease and diabetes. Another area where it applies hybrid animal cloning. Scientists have achieved cross species cloning by introducing genetic material of one species into the egg of another, resulting in cloned animal with the mixed genetic characteristics. For example, in 2003, researchers successfully cloned a mule, a hybrid animal resulting from the combination of horse and a donkey. Hybrid animal cloning has a potential to produce animal with beneficial traits and characteristics for multiple species, while these milestones demonstrate progress made in cloning research. It is essential to consider ethical and scientific challenge associated with these advantages and advancements. Continued exploration, refinement of a technique and ethical consideration will be the crucial of responsible development application of cloning in the future. In continuation, let us also try to understand the various myths and misconceptions that are related with the cloning. The cloning is a subject that often elicits a range of myths and misconceptions due to its association with science fiction, limited understanding and sensitization media coverage. Here are some common myths and misconceptions regarding cloning. The clones are exact duplicates. One of the most prevalent misconception is the clone that are identical copy of original organism. While cloning which produce individual with the same genetic makeup, environmental factor, epigenetic influence can result phenotypic differences, including variation in the physical traits and behavior. Second misconception, cloning create immortal beings. Another myth is that clone organism possess immortality or extended lifespan. In reality, clone have the same natural lifespan as any other individual of their species and are subjects to aging and normal biological process. The third myth is related to bring back to the dead. Cloning is often inaccurately associated with concept rescuing deceased individual. However, cloning only involves replication genetic material, not the restoration of consciousness or memories associated with the original organism. The fourth myth that related that state about cloning is widely used in human. Human cloning is often sensationalized popular media, leading to the misconception that it is a common or widespread practice. In reality, human cloning is highly controversial, ethical debated and largely prohibited by international laws or banned in certain countries. Another point is cloning is the same as a genetic engineering. Cloning and genetic engineering are distinct processes, although they are both associated with manipulating genetic materials. Cloning involves replicating genetic material to create an identical copy, while genetic engineering involves modifying species genes within an organism's DNA. The sixth myth related with the cloning is cloned animal always have health issue. While it is true that cloning can lead to the health issues and abnormalities, it does not mean that all cloned animals have all these problems. With advancement in cloning techniques and improved understanding, scientists have been able to reduce the frequency of health issues and improve the overall health of a cloned animal. Next myth is cloning allow for the replication of intelligence or talent. Cloning an individual does not mean replicating their skills, intelligence or talent. These traits are influenced by a combination of genetic factor and environmental influence, making it unlikely for clones to possess identical abilities or skills. It is important to dispel this myth and misconception to have a more accurate understanding of cloning. Cloning remained complex and evolving field of study with various ethical consideration and practical limitations. Clear understanding of the scientific reality surrounding cloning can help facilitate informed discussion and decision making around its potential application and applications. So, future of cloning, we can say future of cloning hold both possibilities and challenges as scientific researchers advancement and ethical consideration continue to evolve. Here are some points which we can say. Advancement in a medical research, where cloning technique, particularly therapeutic cloning, hold potential for advancement in a medical research. Cloning could help in producing specific tissues or organ for transplantation, disease modeling and drug testing. This application could contribute personalized medicine and development of new treatment for various conditions. In regenerative medicine, cloning may play a role for providing a renewable sources of stem cell for transplantation and tissue regeneration. This could potentially revolutionize the field and lead to breakthroughs in treating injuries, degenerative disease and organ failure. In the area of preservation and endangered species, cloning could be aided in the conservation of endangered species by creating genetic replicates. Effort could be made in revive endangered species or restore ecological balance through carefully managed cloning program. In the field of agriculture improvement, cloning techniques may be employed in livestock breeding to produce genetically superior animal with desirable traits such as disease resistance, improved meat or milk production or enhanced agriculture productivity. Cloning could be contribute to agriculture advantage and food security. In the another way where ethical and societal consideration is also required, the future of cloning will involve ongoing discussion and consideration of ethical, legal and social implications. Societal attitudes towards cloning, perception of human dignity and concern about inequality, commodification and playing the nature with continue to shape the direction ethical framework of cloning practices. For most important area where we can say technological advancement, where advancement in the cloning techniques such as refining the efficiency of the cloning process, reducing genetic abnormalities and improvement of overall human being cloned organisms are likely to be area of a future focus. It is important to note that future of cloning is subject to scientific advancement, regulatory framework, public opinion and ongoing ethical debate, striking a balance between the potential benefit and mindful consideration. So, with this discussion we can finally conclude. In the present lecture, we discuss about the concept of cloning, its historical background, definition, type of clonings, advantage, disadvantage related to cloning, its application, concern related to human cloning, legal restriction on cloning, challenges, limitation, major breakthroughs and myth and misconceptions related to the cloning. In future prospect, along with this, the major key takeaway from the present lecture, we can say cloning raises ethical concern and challenges related to human dignity, autonomy and identity. Legal status varies with the reproductive cloning, generally illegal or highly regulated, and therapeutic cloning subject to different regulation. Cloning faces challenges and limitations including low success rates and genetic abnormalities in the clones. Breakthroughs have been made in cloning animals, stem cell research and other applications. Certain cloning myth misconceptions should be dispelled for a better understanding of the field. The future of cloning depends on a scientific advancement, ethical consideration and social attitude. So, these are the certain list of references through which you can check the information in details. So, with this I thank everyone for your kind attention. Hope this lecture has been fruitful for you. In the next lecture session, you will be able to learn about applications of cloning in details. We will meet you in the next lecture session very soon. Till then, thank you.